So now we know the difference between the individual and population, and we have some basic idea of the population. So here in this lecture, I will be discussing about the genetic structure of population. So how a geneticist will explain about population. So a geneticist describe a population with its equilibrium genetic structure. So this is very important point. The equilibrium genetic structure. We will be seeing this equilibrium genetic structure in detail in later classes. But just for now, you should know that a geneticist will always describe a population with its genetic equilibrium structure under random mating, assuming no sampling variance. The important condition is that there should be random mating, and in that random mating, sample variation should not be there. So I think you know very well what is sampling variation. Sampling variation means suppose we want to purchase 1 kg of potato, and we went to market. There we saw a heap of 100 kg of potatoes, and you randomly took 1 kg of potato out of those 100 kg of potato, and by chance, due to sampling variation, you got all the 1 kg of potato of bad quality, or that suppose it was rotten. So you got all 1 kg of potato rotten, although the heap of 100 kg of potato was not that much rotten, or it was actually a good quality of potato, but by chance you got all the bad quality of potatoes. So that is called a sampling variation. So a geneticist will explain the genetic structure of population at genetic equilibrium stage, and by considering two things. First, that there should be random mating, and there should not be any sampling variation. That means sampling variation, there should not be sampling variation means, the transmission of gene from one generation to next generation is actually a random procedure. So if two genes are coming together, together, then it is merely due to the chance or due to the sampling variance. So the geneticist always explain population assuming that there is no sampling variation. That means all the genes has been transmitted from one generation to next generation randomly. The population property can be described by population parameters. So you have to know about the population parameter. What are the population parameters? And that population parameter will explain the population for a given period of time, and that too it will be changing over the space. So what will be the population parameter estimated here in India? That the same will not be true for the population of Africa or population of Europe. So the population parameter will be fixed for a particular period and for a particular geographical area. Then the population parameters are also trait specific. The type of traits, it will depend on the type of traits. The type of traits that we have already discussed when we were discussing about the statistics. In that few classes, I have discussed about the quantitative and qualitative traits. Quantitative or qualitative traits. So traits are of two types, qualitative and quantitative. Now, the qualitative or Mendelian traits. The qualitative or Mendelian traits are governed by many genes. Actually, it is not many, but these genes may be either one or two or few in numbers. So they are not controlled actually by only one gene, but they may be controlled by one or gene or two gene or few genes. So a population for qualitative traits can be specified by genetic structure for the locus affecting traits in terms of gene and genotypic frequency. So what the population for qualitative trait, it can be specified. Suppose you have to specify a particular population for a particular qualitative trait, then how you will explain, how will specify that population? You will be specifying that population by calculating the gene and genotypic frequency of which gene? The gene which is controlling that particular qualitative trait, and gene will be residing on the locus. So you have to identify the locus that is associated with that particular qualitative trait, and then on that locus you have to find the gene frequency and ultimately genotypic frequency. So the genetic structure of a population is given by specifying the number of loci affecting a trait and series of gene and genotypic frequency that already I have told you. Now, in this way, you can understand that array of gene frequency. You are having a array or series of gene frequency. That series of gene frequency, or you can also say it as a allele frequency. The series of gene frequency or allele frequency, they can be explaining the genic structure of population. Whereas the series of genotype, this will be explaining the genotypic structure of the population. So series of gene frequency will give an idea about genic structure of population, and genotype frequency will give idea of genotypic structure of the population. So what is array of gene frequency? What are the arrays of gene frequency? So array of gene frequency, the number of allele present at a locus. So suppose there is one locus, and one locus, suppose you are having the tallness, gene for tallness, tall plant, short plant, you have studied it in Mendelian genetics, that tall plant and short plant. So suppose this is one locus, and here in locus generally two alleles reside, one coming from mother and another coming from father. So this is, suppose this was the condition, the capital T and small t. So what will be the array of gene frequency? The number of alleles present at a locus affecting the character. The number of alleles, suppose we are trying to find out the gene frequency for capital T. So number of alleles present at a locus. So present at a locus is 1. And the frequency of different alleles at each locus. And what is the frequency of different alleles? How many total alleles are there? So how many total alleles are there? So there are 2 alleles. The 1 by 2 is equal to 0.5. 0.5, this is the array of gene frequency. Similarly, genotypic frequency, array of genotypic frequency or genotypic array. The number of genotype affecting a trait. Number of genotypes affecting a trait. Suppose there are 2 alleles. So these 2 alleles will combine with each other and form 3 genotypes. And out of these 3 genotypes, suppose there are total 100 individuals. So those 100 individuals will be distributed among these 3 genotypes. Now you want to know the genotypic frequency of a particular genotype. So what you will do? Suppose there are 30 individuals with that particular genotype. 30 individuals with that particular genotype. So 30, suppose the genotype is capital T. So 30 individuals with capital T, and total individuals are 100. So 100 divided by 30, that is 0.3. So 0.3 is the genotypic frequency of capital T. So this is how we are finding the gene frequency and genotypic frequency. This I have discussed. I have to discuss in detail in the later slide, in next slide I think. So in nutshell, actually in this lecture I have to discuss about how to explain the genetic structure of population. And the crux of the matter is that the population structure can be explained in terms of gene frequency and genotypic frequency. But the thing is that you have to identify what are the genes or what are the alleles which involved in control of that particular trait, and how many genotypes are there, how many alleles are there present in that on that locus. So these are the things that we have to identify, and the main thing is that, and this has been explained in various ways. So the gene frequency, or it is otherwise known as allele frequency, is the proportion of a given allele in a pair of alleles. Well, when at a locus only there will be only 2 alleles. Even if a particular trait is controlled by a gene which is having more than 2 allele, more than 1 allele or more than 2 allele, having 5 alleles. Suppose a particular trait is controlled by more than 5 alleles. So there may be 5 alleles, but at particular locus there will be only 2 alleles. At particular locus there will be always there will be only 2 alleles. So the gene frequency or allele frequency is the proportion of a given allele in a pair of alleles at a locus to the total number of alleles at that locus in a population. So in this way you can find out the gene frequency or allele frequency, and this can be given by the proportion of given allele in a pair of alleles divided by total alleles present in that population at that particular locus. Similarly, genotype frequency we have already discussed is the proportion of particular genotype among the population. So this is about the genetic gene frequency and genotypic frequency. Now you can see how we are denoting, how we are representing the gene and genotypic frequency in a hypothetical population. Suppose the gene frequency is represented as pi, whereas the genotype frequency is represented as pii for homozygote and pij for heterozygote. So you have already studied that on the basis of composition of alleles, genotype can be either homozygote or heterozygote. In homozygote also, there may be dominant homozygote or recessive homozygote. So here, suppose the gene frequency or allele frequency is pi and genotype frequency is pii for dominant, maybe dominant or maybe recessive, and pij is for the heterozygote. Here in this table you can see the one side it has been the frequency has been given, and another side the genes has been explained. So suppose there are two genes, two alleles. Actually, what about genes and alleles can be used interchangeably. The A1 allele and A2 allele, A1 allele and A2 allele, and you suppose the frequency of A1 allele is P1 and A2 allele is P2. This can also be represented, the frequency of a particular allele can also be represented in the terms of P and Q. This is just a notation. You can represent it by any alphabet or any anything, any symbol, any alphabet or any letter from different languages. Similarly, the genotype, you can say that there will be three. If there are two alleles, there will be three genotypes: A1A1, A1A2, and A2A2. A1A1, A1A2, and A2A2. So the A1 frequency of A1A1 genotype will be P11. For A1A2 genotype, it will be P12. Frequency will be P12, but it will be multiplied by 2. Why it will be multiplied by 2? If you remember, just try to remember the Punnett square. Punnett square, you are seeing that a Punnett square is the way of representation of different gametic combination. So here we were putting the male gamete and here female gamete. So suppose there are two alleles, capital T and small t, capital T and small t. So here you can see the capital T, then capital T, capital T, small t, and small t. You can see the heterozygote genotype. Heterozygous genotype, they are actually not single genotype, but they are double. That is why because there will be different combination of male and female gametes. So that is why in heterozygote we are multiplying it by 2. Similarly, the recessive homozygote, the frequency is P22. And these frequencies, this genotypic frequency can also be given in terms of DHR. D because of dominance, or you can write anything depending on your assumption. So D is here, it has been represented because it is dominant, H because it is heterozygote, and R because it is recessive homozygote. Now, the one condition at equilibrium population, when population is at equilibrium, what is population and equilibrium, that I will be discussing in the next class. Just now you remember that P plus Q, what is P plus Q? P is the frequency of allele A1 and Q is the frequency of allele A2. So P plus Q. So there are only two alleles at a particular locus. So P plus Q will be equal to 1. The total allelic frequency will always be equal to 1 at equilibrium. So P plus Q is equal to D plus H plus R. What is D plus H plus R? So D is the frequency of dominant homozygote. H is the frequency of heterozygote and R is the frequency of recessive homozygote. So D plus H plus R, the frequency of all the homozygotes at a particular in a particular population at equilibrium will always be 1. So this you have to remember that P plus Q is equal to D plus H plus R is equal to 1. And with this equation you can find how this P frequency of allele A1 can be given by D plus half H. Why D plus half H? Why D plus half H? Because in heterozygote you will get only one A1 allele, only one A1 allele. Another will be A2 allele. So that is why we are taking half of the heterozygote frequency. Similarly, Q is equal to R plus half H. R plus half H. Only we are taking half of the heterozygote frequency. Then now just see one example of genetic structure of population for a single locus. Actually, this all we have discussed. What example I have discussed just now? I have discussed the example that is also for the single locus. So we'll just again repeat it so that you can clearly understand it. What is the gene frequency and genotype frequency, and how we can find out the gene and genotype frequency. So two alleles at a locus. When there are only two alleles at a given locus, they may be denoted as either A1 and A2 or capital A and small a. You can represent it in any way. The respective frequency will be denoted by P and Q. So we all know. In diploid population, two alleles are at a locus. I have already discussed that there will be two alleles, one coming from mother and another coming from father. So in diploid population of n individual, there are n individual in the population with A1 and A2 alleles in combination. There will be three genotypes. What will be the genotypes? A1A1, A1A2 and A2A2. And this can be represented, this whole statement can be represented as, suppose there are three genotypes, A1A1, A1A2 and A2A2. So A1A1, the number of individuals, suppose A1A1 type, there are N11 individual, heterozygote, there will be N12 individual, and homozygote recessive N22 individual. So what will be the genotypic frequency? So total number of individuals are N. So genotypic frequency of A1A1, what will be the genotypic frequency of A1A1? So it will be N11 divided by N. And similarly, you can find the genotypic frequency in terms of gene frequency or allele frequency. So P11 is the genotypic frequency. It can be given by P square. Why P square? Because in P11 there will be two similar alleles, A1, A1. That is homozygote genotype. So in homozygote genotype, you will see the frequency is P square, and P square can be given as D, that the dominant. Similarly, 2P12 is equal to 2PQ, that is heterozygote, and P22 is equal to Q square, that is equal to recessive frequency of recessive genotype. So this is how the gene frequency and genotype frequency can be represented. Now the next, in the next slide, here you can see the three genotype A1A1, A1A2 and A2A2 can be identified phenotypically. So these three genotypes, if you remember the Mendels law or Mendels monohybrid ratio, you can see the in Mendels monohybrid ratio, the phenotypic ratio was 3:1. 3:1. Why it was 3:1? Because if you remember the Punnett square, these three will appear similar. Phenotypically, these three will appear similar. Only homozygous recessive will appear differently. So, rightly it has been said here that the three genotypes A1, 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 A2 and A2, A2 can be identified phenotypically only when the inheritance pattern is intermediate. So why it is? Because these three individuals you cannot differentiate phenotypically. Why? Because A1 is dominant over A2. So only and only if A1 and A2 are not showing dominance on each other, then only these three genotypes will appear differently. Otherwise, the ratio will always be 3:1. Now, the number of individuals, the three phenotypes are counted and converted into proportion among the total individual known as genotypic frequency. So, what we are talking here, we are here uh, talking that there is no dominance. There is no dominance, phenomena of dominance. So, what we are considering that A1, A1 and A1, A2 genotypes are different. They are not, they are phenotypically different. So, how to find the gene frequency or allele frequency PA1? What will be the frequency of A1? So, frequency of A1 allele will be number of A1 alleles divided by total number of alleles in that population. So, number of, what is the number of A1 allele? It is A1 allele, from where A1 allele will come? A1 allele that will come always from homozygous dominant or homozygous, one homozygous and half of the heterozygous. Why homozygous? What was the composition of genetic composition of homozygous? A1, A1. So from there, two A1 allele will come. And from heterozygote, the heterozygote frequency was 2A1, A2. So one A1 allele will come from there. So you can represent it as 2 into N11 plus N12 divided by total number of individual. Total number of individual was N and the number of alleles possessed by those N individual will be 2N. So this is how we are finding out the frequency of A1 allele. Similarly, you can find the frequency of A2 allele, the number of A2 allele divided by number of total alleles. And from where A2 allele will come? A2 allele will come from the homozygote, the recessive. If you remember the recessive homozygote, the two alleles will come from there, and from heterozygote also you will get one allele. So the formula will be 2n1 n2 n2 plus n12 divided by total number of individuals, and this will be equal to R plus half of the heterozygote, or here it was D plus half of the heterozygote. And now you know the P plus Q is equal to 1. Therefore, P is equal to 1 minus Q and Q is equal to 1 minus P. So, even you find out the allele frequency of one allele, or frequency of one allele when there are two alleles. If you know the frequency of one particular allele, you will get the frequency of another allele, because at equilibrium, the frequency of sum of the frequency of all the alleles will be equal to 1. So this is how we are specifying or describing a population population in terms of genetic structure, in terms of gene and genotypic frequency. So this is all about the genetic structure of population. In the next class, we will see about the history of population genetics. We'll see some of the scientific contribution, some of the scientists who has contributed in the development of population genetics. Thank you.